Okay, well, we're all familiar with directional environmental change and the problems it creates for many, many species, and the typical response historically has been range shifts, but a variety of factors such as habitat fragmentation means that many species can't shift their range. So they basically, if they're going to avoid extinction, then they really have to adapt to the changing conditions in situ. And this raises a general question of what determines the likelihood that effective long-term adaptation can occur. And then a question that's been raised more recently is whether long-term adaptation is aided by phenotypic plasticity. So I'm going to just go over very quickly the first general question. Haldane was really the first person to seriously address the issue of what limits the rate of adaptive evolution in his famous paper on the cost of natural selection in 1957, where he argued that because natural selection in driving the change of an allele frequency requires a genetic death, as he expressed it, that this limits the rate at which adaptation can occur. And with a bit of hand-waving at the end of this otherwise very algebraic article, he estimates that maybe there's a maximum rate of about 1 substitution per 300 generations. Many years ago now, I did some individual-based simulations suggesting that long-term adaptive evolution can be much faster in large populations, provided that the input, well, in a large population, the input of new beneficial mutations per locus will be relatively high. And this is going to be designated by M, and it's actually equal simply to twice the carrying capacity times the beneficial mutation rate per locus. And provided you're over a barrier of about M greater than 0.1, then adaptation can proceed pretty rapidly. The problem is, if that's not true, then populations get delayed. And in small populations, and in this context, small population means less than tens of thousands of individuals. The rate drops off rapidly because obviously M is dropping, because the carrying capacity is dropping. Okay, so we can ask, so can phenotypic plasticity actually sort of help save small populations? The idea being that phenotypic plasticity allows populations through the individuals to essentially buy time for a population subject to directional selection, as it were, waiting for mutations to come along. The prior work on this was a really nice paper by Chauvin, Landy, and Mace in 2010, and did a quantitative genetic model including these things, the directional environmental change, plasticity, and their metric, which is the same as I'll be using, is the extinction of populations. How fast can you change the environment to avoid extinction? Just to get you in the thinking about phenotypic plasticity, reaction norms are the relationship between environment and a trait, so this would be the reaction norm of a single genotype. And the thing to remember is, of course, the fitness of that genotype varies along that reaction norm. So to understand this, you then have to create an adaptive landscape where you've got a fitness dimension as well. And a nice way of summarizing this is what's often called the tolerance curves, which is the relationship between the environment and the fitness. And with environmental change, of course, you imagine the environment is sort of moving along underneath the genotypes. So in this context, if you imagine a population that was fixed for the genotype, which has the red tolerance curve, as the environment moves away from its optimum, then eventually it's going to get to a point where this population could not replace itself and will go extinct. I'll add one other thing. The dashed line here is reflecting when you have plasticity represented by the variation in this tolerance curve, where plasticity has reduced proportionately all of the fitness variables. Back to that later. Okay, so what did they find in their analysis? And what they found was the fact that this is the rate of environmental change is sort of increasing as you go up here. And obviously, as you increase the rate of environmental change, the risk of extinction is going to increase. But when you have no-cost plasticity, then you find a monotonic increase in the likelihood of persistence as plasticity goes up. So plasticity in this context is good. It's allowing population to persist. These other curves I wouldn't worry about are related to when you have cost of plasticity. Okay, so I was interested in adding plasticity in the model that I'd used to examine Haldane's cost, just to see if I got the same basic result. And the individual-based model that I'd used in that previous study, it had all of the various components. It had directional environmental change. It had population dynamics built into it with density-dependent regulation. And of course, it had an adaptive trait determined by n loci at time. When the environment has moved on, the best genotype to be has all the 1 alleles, and then so on. You then want all the 2 alleles, and so on and so forth. And the rate of environmental change is measured by the number of generations between each of these particular cycles. So in other words, if going from all the zeros to needing all the ones, that takes a long time, then obviously environmental change is very slow. On the other hand, if it's very fast, environmental change is very rapid. Okay, so how do we add phenotypic plasticity into this? And simply, the easiest way is just to broaden the fitness curve, which we can now refer to as the tolerance curve. And so you shift from this being relatively non-plastic to this situation where you just widen these, in fact, in this particular case, by 3-fold, to give an individual genotype the ability to survive over a much wider range of conditions. And the question is, with this model, with this simulation model, does plasticity still promote population persistence as found in the previous study? And the answer is that it doesn't. Just to confuse you, the scale on this is round the wrong way, so environmental change actually, as you go down, is getting faster and faster because it's the number of generations between these 2 points. So this is a slowly changing environment, this is a rapidly changing environment, vary the number of loci, this is the input of beneficial mutations, but the pattern is very consistent, and that is, except when you have only 1 or 2 genes of major effect, having plasticity, which is the filled-in circles, is always worse than limited plasticity. So the more plasticity you have, the worse you are. In other words, these guys could not survive the faster rate of change that these guys can survive. So we got the opposite result. Obviously, the question is, why have we got the opposite result? This is not just this is a qualitative difference. It's not a quantitative difference. But when you look at the models, even though they're expressed in very different terms, they're actually essentially identical, except for one thing, and that is how how additive genetic variance is treated. The Leonard and et al. Paper uses a conventional form where the trait value of genotype I is related to the breeding value measured in some reference environment plus the effects of plasticity, and that's a traditional way of doing it. But I was very worried about this because that's fine when you have a limited range of environments, but here we're talking about a moving optimum, moving further and further away from any reference environment that you may have set up. And so what that suggested was that it's going to drive an association between genetic variance and plasticity that didn't seem to be appropriate. So I used a different approach, which is to use the baseline as the optimum of each genotype plus an effect of plasticity, which of course I defy anyone to really understand simply, so I tried to make an incredibly complicated graph to try and illustrate this. So this is the incredibly complicated graph, and I'll try and walk through this. So environment is down the bottom, the trait value is going up the y-axis. Let's start with a red line, which links in a particular environment. We can look at the red one there. It says that in this environment, this is the best optimum trait value. Okay, so just linking them straight along there. Okay, so now what we say is, so the Leonard et al. Paper uses a reference, the environment at t equals 0, which will be this environment here. So what we imagine, if we have something with the green reaction norm, that's what that is. So the green reaction norm of this particular genotype, we take it back to the reference environment and we mutate it. We add some genetic variance, additive genetic variance, so it crawls up the axis to this point to the next green line. We then project that back to determine the optimum, and the optimum is E2. We then look at a situation where there's actually more plasticity in the population, which is the blue line, so it's a steeper reaction. We do the same thing, we project it back to our optimum, and we find its optimum is E3. The point that I need to make here is that the effect of a fixed mutation is varying in its effect on the optimum environment depending upon how plastic the population is. So you're getting a combination of just the standard trait variance and the plasticity. And I felt that this was inappropriate, so instead I wanted to model it so that if you had a mutation on this trait value, you increased it by D again, and then you went just straight back to this trait optimum line, and that will give you an environment E1, so that any mutation in any population of that size is going to cause the same magnitude of change in its trait optimum. What's happening in the alternative model, which I think is driving the result, is the fact that the effect of a mutation is giving you more, if you like, adaptive bang for the buck as plasticity increases. So if there was no plasticity, you'd actually go to here. With some plasticity, you go to there. With a lot of plasticity, you go to there. And I think this is actually driving this advantage of plasticity that Chauvin et al. Actually found in their model. Okay, so now the question is plasticity individually advantageous? So I wanted to simulate a single locus determining plasticity and also the possibility of having a cost. And I'm not going to go into how I did the cost, it's a simple multiplicative effect. So what we have here is a graph that shows a simulation of about 2,500 generations. And this is where you have a high cost of plasticity. The result of having a high cost of plasticity is plasticity does not evolve. And what happens is the optimum genotype at this point is that black line. And you see that the blue bar represents where this population's got to. It's tracking the environment incredibly well. As you decrease the cost of plasticity, you begin to actually favor plasticity, but the tracking of the environment goes down and down. And this can become a problem under some circumstances. So in this particular case, put my finger over it. So this is just a couple of simulations to illustrate the point. Here the rate of environmental change is 50% of the maximum that it could be for a non-plastic population. When the cost of plasticity is high, so that essentially plasticity does not evolve, this population does well. The lag is very small. This is in terms of allelic units. The lag is very small, plasticity doesn't evolve. On the other hand, if the cost is small, this is basically, as it showed before, is the lag gets large and the plasticity builds up. The problem is, if you then increase the rate, in this case to 80% of the maximum, when plasticity has a high cost, it doesn't evolve. But when it has a low cost, the consequence is it evolves, but all the populations go extinct. So we can then quickly summarize. So in large populations, they can adapt very rapidly, but small populations are severely limited by a slow input of beneficial mutations. Phenotypic plasticity that increases tolerance is individually beneficial, so it's always favored if there's a low cost. But over the long term, it slows adaptation and can cause extinction. Thank you very much.
Feel free to ask a question while we're changing those.
Yeah.
So it seems to me that you never allow the reaction norm to exactly equal that tolerance, the optimal curve, right?
Right.
So,
well, I mean, you could, but that wouldn't. So you'd have perfect matching.
right. So it's not that plasticity can't. It's you've artificially constrained the range of plasticity.
Well, I mean, I actually don't. So when in those ones where we allow it to evolve, in some cases it's getting to a bigger and bigger number, which actually means the tolerance curve is getting wider and wider and wider and wider, so it's flattening out. So basically, the population become, they can track the environment perfectly, but the thing is that selection can't work at that point.
Okay, next up we
Because
have
any
Leonard.
genotype, one genotype